Welcome to this lecture on biomedical data, types and challenges. This lecture will discuss big data, types of biomedical data, and the challenges in the handling and analysis of biomedical data. Let's start with the topic of big data. We are living in times of a medical data explosion. One estimate suggests that medical data is expected to reach 10,000 exabytes by 2025. As seen in this table, if you're familiar with terabytes, a petabyte is over 1,000 terabytes, and an exabyte is over 1,000 petabytes. Estimates have seen the expansion of medical knowledge exponentially increasing throughout the previous decades, where in 1950, knowledge was viewed as doubling every 50 years. In the 80s, it was seen as doubling every 7 years, and in 2020, it was doubling every 73 days. Healthcare data, in particular, is projected to experience a 36% compound annual growth rate through 2025, which is much faster than industries such as manufacturing, financial services, or media. Data of all types has been discussed via characteristics referred to as the V's of big data. The first of these is data volume, which describes the sheer scale of data in terms of storage size and units of bytes like petabyte or exabyte. Next, we have the velocity of data, which describes the real-time streaming of data or speed of data generation. It's fairly easy to imagine how quickly data can be generated in real time from technologies in healthcare such as bedside monitors and wearables. Next, we have the variety of data, which simply gets at the many different forms in which data can take, from electronic health record text to clinical images, genomic sequencing information, and many others. These were the first of 3 V's described in the literature with respect to big data. However, other V's have since been proposed that can be important for consideration. For example, we have the veracity or validity of data. This characteristic describes the truthful quality of data versus the potential for uncertainty in data. It's here that we're concerned with the possibility of data being noisy or containing biases. Furthermore, we have the value of data. This characteristic asks what we can actually do to the data in hand. Is it useful or relevant to a given problem or set of problems? For example, is given data going to be useful for predicting an outbreak before it happens? And lastly, we have the volatility of data. This gets at the permanence or persistence of value in given data. This can be about how long certain types of data are retained, changing relevance to a specific application, or changing validity as new data becomes available. From a clinical and biomedical research perspective, we have begun to see a shift from small data to big data. We can think of small data in the context of a patient's chart, which is a snapshot in time where the doctor saw a patient who came in with an illness. In this context, research is primarily hypothesis-driven, where a doctor forms a theory and orders specific medical tests to confirm it. Here, the data volume tends to be smaller, on the scale of megabytes, and focuses at a lower resolution, such as the organ or system level. Overall, this approach is reactive, where a doctor seeks to treat the symptoms at hand, and the scope or diversity of their patient experience may be largely limited to a single clinic or hospital. The traditional tools of small data research combine human intuition with traditional statistical analysis. Differently, we can think of big data research as focusing on information such as population-wide genomic analysis, where the data gathered may be longitudinal, multidimensional, and multimodal. Research here can be more data-driven, also known as hypothesis-generating, where tools like artificial intelligence can be used to mine data and find hidden associations that no human being may have noticed. Here, the data volume tends to be extremely large, on the scale of terabytes to petabytes or beyond. Such research can also focus on much higher resolution information, for example at the molecular level. Also, there's a shift from not just reactive to predictive medicine, where we seek to prevent or limit the severity of future disease. This type of research shifts more into drawing data from a global level, for example across diverse ethnic and genetic backgrounds. Increasingly, the tools for this type of big data analysis have shifted into the realm of artificial intelligence and machine learning. One of the major goals of big biomedical data analysis is to convert raw, messy observations into actionable computational knowledge. This newer data-driven or hypothesis-generating approach is overall less dependent on prior knowledge, and thus can have a greater potential for novel hypothesis discovery. Probably not surprisingly, the rise of these hypothesis-free methods were initially greeted with a polarized mixture of overblown enthusiasm and complete distrust, as some people believed that a priori hypotheses were no longer necessary, while others argued that these new approaches were an irrelevant distraction from established traditional methods. However, these days it's fair to say that hypothesis-generating approaches are largely recognized as not only being synergistic with traditional methods, but also being dependent on them. After all, once generated, a hypothesis still needs to be tested. Here on the right, we see an illustration of the synergistic cycle between hypothesis-driven and data-driven experimentation. One of the main reasons there's been such interest in shifting focus to big biomedical data research is the goal of achieving precision medicine. Precision medicine is an innovative approach to healthcare that tailors disease prevention and treatment to the individual characteristics of each patient. In short, it's about finding the right treatment for the right patient at the right dose and at the right time. For example, when it comes to treatment, the old way might have been to say that if 100 people have the same type of lung cancer, we will give them all the same chemotherapy, and if it works for 60% of them, we consider that to be a successful treatment. Alternatively, a precision medicine approach to treatment might see that those 100 people actually have 5 different genetic subtypes of cancer when using population-scale genomic data. This might allow us to identify the specific 20 people who respond to a targeted drug and the 10 people for whom the drug would be toxic. One possibly idealistic vision of precision medicine is to move away from the small data approach of traditional randomized control trials, which focus on the average patient, and instead move towards big data approaches that enables what's been referred to as an N-of-1 clinical trial, instead focused on finding a treatment that works best for the individual. As we can see in this figure, the surge in research focused on big biomedical data started kicking off between about 2012 and 2015, and so far it seems unlikely to slow down anytime soon. And in this figure, you can see some historical landmarks in the evolution of how biomedical fields have come to embrace big data analytics. Here, the birth of big data has been attributed to some of the first reports of data analysis by John Graunt during the London Plague in 1663. After this, the innovation and invention of several hardware processing and storage devices paved the way for managing new data to be generated. Later, the emergence of computational languages, along with database management systems, aided in the global evolution of information technologies and big data analytics. Now, we'll move on to look at different types of biomedical data specifically. Raw biomedical data can come in many sources and many forms. For example, it could come as clinical notes, test reports, electronic health records, or photographs such as X-rays or MRI scans. Some data may already be in a digitally accessible form, for example in a database, but many sources of biomedical data start in a non-digital and unorganized form. Such data will, of course, first need to be appropriately digitized and organized for use. Now let's review some common data types. First, we have tabular data. This is a data format most of us would be used to, involving rows of instances and columns of features. Next, we have a directed graph, or a network. For example, this is often how ontologies of terms are arranged to express hierarchies or categorizations. We could also have images, or videos, which are simply images with the added dimension of time. Quantitative data can also be measured over time, which is generally referred to as time series data. Examples of this might include patient heart rate or electrocardiogram measurements over some period of time. And our last example is text-based data, which is often unstructured and might require the application of natural language processing techniques to extract useful structured information. Most any data sources could be extracted into a form of tabular data, which is the most conventional format to use for traditional statistical analysis and/or machine learning. One basic but important categorization of data types is that of structured versus unstructured data. Structured data is often what we refer to as tabular data, where the information can be displayed across rows and columns, as well as within relational databases. The basic unit of information is generally numbers, dates, and short strings of text. Generally, the availability of structured data is more limiting, since it requires careful data collection and/or data processing to assemble it. But in general, it requires less storage space and is easier to analyze using traditional and a wider variety of modern machine learning and AI techniques. On the other hand, unstructured data is a catch-all for pretty much every other kind of data out there, including images, audio, video, time series data, word processing files, emails, etc. In short, unstructured data is simply data that cannot be easily displayed as rows or columns, or within relational databases. Most of the data out there in the world would generally be considered unstructured, and this type of data typically requires a lot more data storage. One important consideration for data science is the variable types. Understanding variable types is important for selecting the right analysis methods and ensuring respective variables are treated appropriately. Let's quickly review different variable types. Here, the term variables can reference either features or outcomes, where features can be thought of as data variables that we can examine to search for patterns or associations, and outcomes are the data variables that we may want to make a decision or prediction about. For example, does the patient have an infection or not? First off, a variable can be numeric, otherwise known as quantitative. For example, this could be the age of a patient. Numerical values might be continuous valued, appearing anywhere within a numerical spectrum, or they could be discrete, for example, only appearing as integer values. Going back to this age example, we know that true age is a continuous value. However, in patient data, age is usually captured as a discrete integer. Next, a variable could be categorical, otherwise known as a qualitative variable. For example, this could be something like patient race. These variables have no intrinsic ordering to them. Of note, for many machine learning algorithms, categorical variables as features present an extra challenge. Specifically, most algorithms train models under the assumption that all variables have some quantitative ordering. One way around this is to re-encode categorical features using something like one-hot encoding, which breaks a single categorical feature into a set of new features describing each possible category. And lastly, a variable could be ordinal. For example, this could be something like tumor stage. These variables are similar to categorical ones in that they often appear in data as text entries, such as stage 1 or stage 4. However, these values can be re-encoded as numerical values with a quantitative ordering. Notably, there are generally some assumptions that are being made when we numerically encode ordinal variables. For instance, how far apart are the different groups? After encoding, these variables can be treated as discrete numerical variables. Also of note is that discrete variables with only two values are referred to as binary, and we generally encode these with a 0 or 1 value. Categorical variables with only two values are typically called Boolean variables, described with the values true or false. Here we can see the results of a COVID-19 test, which might be labeled as positive or negative in a given dataset. It could also be labeled as true or false, or encoded with a 0 or 1. Variables with only two possible values are easier to deal with since the order between them is pretty much arbitrary, and they can all be treated as either quantitative or categorical in data analysis. Having reviewed feature types, let's look at an example of some tabular data. The rows here represent individual instances with a header at the top. The header identifies the different columns in the data, or in other words, the names of the different variables in the data. Included in these columns are feature variables, the outcome that our downstream model will aim to make predictions on, and sometimes the data might include an ID, which identifies the unique instances of the dataset. Of note, instance IDs should not be provided as a feature for modeling, as it is a unique value for each row and thus offers no usable information for modeling. Now let's identify some of the unique variable types we discussed in the last slide. The feature age is an example of a discrete variable. Both zip code and biological sex have here been encoded as categorical variables. Body mass index, or BMI, is an example of a continuous variable. And income, as presented in this dataset, is an example of an ordinal variable that also has a known ordering, in this case, low, middle, and high. Depending on the algorithm implementation to be used downstream, we might want to re-encode the values of income with something like the values 0, 1, or 2, corresponding to low, medium, and high. And lastly, we have the feature called insured and the outcome variable called diagnosis, both of which could appear as Boolean or numeric based on how the values were recorded, or whether they were encoded as a 0 or 1. Given the data types we've just covered, let's take a look at some specific biomedical data examples. For instance, visual data, which can include images and video, might be in the form of X-rays, MRI scans, pathology slides, or surgical videos. Unstructured text data might come in the form of clinical notes, discharge summaries, and patient emails. Typical structured or tabular data might come in the form of lab results, blood pressure, or patient age. Temporal or time series data might come in the form of ECG waveforms, EEG brainwave data, or wearable sensor data, such as from a pulse oximeter. Further, we could have acoustic data that seeks to capture patient cough sounds, heart murmurs, or speech patterns. Modern-day biomedical data has a tendency to have a very large number of features, and in some cases, a large number of instances as well. As we discussed, biomedical data can come from many sources, including features and other data streams from the genome, epigenome, proteome, microbiome, microRNAome, electronic health records, imaging, mobile health, and many others. In trying to understand and predict biomedical outcomes, it may be desirable to integrate all available data into a single dataset for analysis. However, this comes with a number of logistical concerns, like data storage and computer memory limitations, as well as analysis concerns, such as the efficiency of feature selection and modeling in the context of having a huge number of potentially predictive features. One major type of biomedical big data is that of multi-omics data. Multi-omics data seeks to integrate various omics data sources, such as genomics, epigenomics, transcriptomics, proteomics, and metabolomics, as an approach to systems biology. A primary goal of systems biology is the study of the whole system and its interactions, rather than just isolated parts. One advantage of this type of approach is that it can help us to separate correlation from causation. For example, a person might have a genetic mutation for a disease, and this could be observed from genomics data. But that gene is never expressed, as may be observed in the transcriptomic data, or the protein is never formed, as observed in proteomic data. And as a result, that patient might never get sick. Multi-omics can help us capture this kind of nuance. Another general advantage is that multi-omics can help capture biological breadth. For example, some diseases like cancer or Alzheimer's are simply too complex to be explained by a single gene, and multi-omics can allow artificial intelligence models to find signatures across different layers of omics data that may signal the start of a disease. Earlier, we brought up the topic of precision medicine as a driving goal of big data analysis. Omics data represents one of the key data pillars driving precision medicine efforts. Omics data represents blueprint information, looking at high-resolution characteristics of a patient. This includes DNA sequences from genomics, gene expression information from transcriptomics, proteins from proteomics, and metabolites from metabolomics. This data is meant to help us understand the biological underpinnings of disease. Another pillar is that of clinical data, which generally represents lower resolution characteristics of a patient. This comes in the form of electronic health records, medical history, lab results, imaging, and documentation of treatment responses. Further, precision medicine can use data from a patient's lifestyle and behavior to provide some short-term context information. This can include things like diet, exercise, habits, and environmental exposures, which can be crucial for understanding chronic disease. And lastly, environmental and social data can offer a longer-term context, including information about where the patient lives and their potential lifetime exposures, such as air quality, community resources, and socioeconomic factors that might impact their health. This figure gives an overview of the variety of data types that might be useful in real-world biomedical and clinical data analysis. Some other data type examples that we haven't really mentioned yet include data about medications, such as types and doses, insurance claims data, family history, social media sources, and the clinical and biomedical research literature itself. One other topic we'll bring up with respect to biomedical data is that of multimodal data. This refers to combined data that comes from multiple modes or formats, such as text, images, audio, video, and structured data, each describing the same entity or event. In the context of artificial intelligence, a multimodal model is one that can see the image, read the clinical notes, and hear the patient's voice simultaneously to form a single comprehensive understanding. It's worth highlighting the difference between multimodal and multi-omics data, since it could be easy to confuse the two. But we can think of them as representing different directions of data. Multi-omics focuses on vertical integration. In other words, it looks deep into the biological layers of a single cell or organism, for example, the hierarchy of DNA to RNA to protein. Multi-omics is entirely focused on biological and molecular data. On the other hand, multimodal data can be viewed as focusing on horizontal integration, tackling the combination of different types of data modes and formats. In the last section of this lecture, we'll discuss some of the many challenges inherent to biomedical data. As we've covered, biomedical data is certainly one major source of big data in the world, which presents inherent challenges. Biomedical data of certain types can have a very high dimensionality. For example, a single genome can have 3 billion base pairs, and a single CT scan can have millions of voxels, where a voxel is the 3D equivalent of a pixel in a 2D image. This creates immediate challenges with respect to data storage, processing, and handling. This can also lead to challenges regarding downstream analysis of the data, and whether we have sufficient computational resources to complete the desired analysis given the scale of the data. Other common analysis challenges include working with diverse data and feature types, dealing with data with an imbalanced outcome, which is a common challenge in classification tasks, and how to identify or how to consider covariates in the data so that we can better separate correlations from causality. Some of the most challenging aspects of data science in general revolve around data preparation, where we have to deal with things such as data missingness, whether or not to consider data transformations, and also there are many challenges in the wrangling of data. One possibly important consideration in data wrangling is how to effectively combine the same data types across different original sources that may come from different institutions, such as hospital systems, states, countries, clinical trials, etc. Here, techniques such as data harmonization, data integration, and data standardization are important but challenging tasks, which we'll talk about a bit more in a minute. Further, another data preparation challenge can include the fusion or combining of multimodal data types for analysis, which we'll also touch on more in a couple slides. Let's start by talking a bit more about data harmonization, which is a task which focuses on converting disparate data into a cohesive, standardized format. This data could come from different EHR systems, different genomic databases, and different clinical trials, as just a few basic examples. The key challenges of data harmonization involve resolving differences in syntax, structure, and semantics. We'll discuss these more in a moment. Data harmonization seeks to create comparability across datasets that may have been originally incompatible. This enables a unified analysis of previously fragmented information, which helps convert raw data into actionable medical intelligence. As we mentioned a moment ago, to truly harmonize data, you must address the challenges of syntax, structure, and semantics. Resolving the syntax of data seeks to tackle surface-level inconsistencies in data formats. It seeks to enforce a single consistent representation for the values across the data. For example, ensuring that all dates follow a single format instead of a mix of formats coming from different data sources. Structural mapping, on the other hand, addresses differences in schema integration, where we seek to create a unified blueprint for the data, defining how different data models relate to one another. For example, mapping a single address field into separate columns for street, city, and state. The goal of structural mapping is to create a single logical hierarchy, as well as to define the rules needed to transform data from all sources into a single dataset. Lastly, we have semantic alignment, which is often the most challenging, given that it deals with the intended meaning of the data. This task seeks to ensure that a concept means the same thing across all data sources. For example, in healthcare, this could mean mapping local lab codes into a universal standard. For example, mapping different clinical terms using the SNOMED CT's terminology standard. This aspect of data harmonization typically requires deep domain expertise to correctly interpret the context behind data. It's also worth taking a moment to distinguish data harmonization from data integration and data standardization. In short, as a metaphor, standardization ensures everyone wears the same uniform. Integration gets them all in the same room. And harmonization ensures they all speak the same language and work towards the same goal. This table on the right gives a bit more detail breaking down the differences between these three different tasks. While all three of these are important to consider, data harmonization in particular is what enables deep insights from complex, multi-source data. With respect to data integration, it's useful to point out a few particular challenges that relate to multi-omics data, which is by definition high-dimensional data. The integration of these different omics sources can be a computational challenge for AI systems. This is in part due to the variety of data, where each layer of the data can have different scales and formats. For example, DNA is a sequence of characters, but proteomics is a list of concentrations. Another common challenge is missing data. It's rare to have all omics layers for every patient in a study, and this can lead to the need for missing value imputation, which is the prediction and replacement of missing values. And lastly, the dimensionality of multi-omics data poses inherent challenges for integration. For example, you might have 20,000 genes, 100,000 transcripts, and 10,000 metabolites for a single person, but ultimately only 500 patients in total for your study. The problem of having more variables than samples can make models trained with AI more prone to overfitting, and introduce other challenges that we'll discuss in a moment with respect to the curse of dimensionality. Before moving on, we'll also take a moment to consider multimodal data fusion challenges, which impact how the downstream modeling of the data takes place. One of the hardest parts of multimodal AI analysis is the fusion of data. For example, how do you mathematically combine a pixel and a word? Thus far, a few general strategies have been described. The first is referred to as early fusion, where we do our best to combine all the data into one big file before an AI strategy starts learning. The second is called late fusion, where we train separate AI models for each data modality. For example, one for images, another for text, and at the end, we might have them vote on a prediction as an ensemble at the end. Lastly, we have the most modern approach, which is a mid or joint fusion strategy. Here, the AI would seek to learn the relationship between the models and data modes. For example, learning that the word fracture in a note usually relates to a specific bright line in an X-ray. Here, each type of data modality is first passed through its own dedicated feature extractor, such as a neural network tailored to that specific data type. And then once these networks have distilled the raw data into high-level concepts, these concepts are fused into a shared representation space. This table on the right further outlines some of the differences between these three general strategies for multimodal data fusion. Now let's pick up with some additional common challenges in biomedical data. Another set of challenges revolves around privacy, ethics, and competition that can all make it harder to work with biomedical data. For example, HIPAA in the United States and GDPR in Europe set strict regulations that govern how data can be used. This can limit access to data, what we can do with it, and where we can store and analyze it. We also need to concern ourselves about protecting the privacy of patients from whom the data is collected. We have to be cautious about working with and sharing data, even if it's considered to have been de-identified or scrubbed of key patient-identifying information. When dealing with high-dimensional data like DNA or facial scans in MRIs, it's possible that patients could be re-identified, even if names or other common identifying information has been removed. Another logistical issue is the data silo problem, largely due to competition between researchers, institutions, and financial interests, as well as concerns about legal risks. Hospitals or other institutions can be reluctant to share data with each other, leading to smaller, more fragmented datasets for research. There are also a number of challenges with respect to detecting signal in data. For example, data can be noisy, which means that even with an optimally trained model, we can never expect to make 100% accurate predictions. On the other hand, it's possible that some or perhaps even all features required to understand an outcome could be missing from the data. As an example of this, imagine you're trying to train a highly predictive model of lung cancer when only genetic data is available, but environmental data, such as pack years of smoking, is not. Additionally, it can be difficult to model the impact of features with rare values. This is common, for example, in genomic datasets, such as whole genome sequencing data or whole exome sequencing data. And further, the relationship between features and outcome can be obscured by complex multivariate associations, which we'll discuss in more depth in the next couple of slides. Lastly here, we have challenges in data and model reliability. Here we concern ourselves with the reproducibility of our results on other future data, or data from another hospital, region, state, or even country. Trusting a model is often paired with the challenge of being able to interpret or explain the predictions of a model. And lastly, we want to identify and account for the possibility that our data or model may have underlying biases in it. Now let's take a closer look at the challenge of complex patterns of association in data. We'll do this from the perspective of genetic data. First, here's a schematic attempting to illustrate some of the possible ways that features on the left can be associated with outcome or phenotype on the right. Here, the word phenotype just refers to some trait of an organism that we're interested in. Features here could be any of the things we've already mentioned. At the top, we have the simplest association between a single gene or genetic feature and the target outcome. This is known as a univariate association. Next, we have pleiotropy, which is when a single gene or genetic feature has an association with two or more seemingly unrelated outcomes. Next, we have epistasis, also known as multivariate interaction. Here, two or more features interact with one another to impact outcome. Notably, in the most extreme case, it's possible for feature interactions to strongly predict outcome, even when none of those individual features have any univariate association with that outcome. And lastly, we have heterogeneous associations. The first heterogeneous association we'll mention is genetic heterogeneity. This is a situation where different genes or genetic features can produce the same or similar outcome in different instances. Phenocopy is a similar situation, where an environmental feature could impact the outcome of instances in the same way as a gene or genetic feature. And beyond any of these individual, more complicated associations, in real life, they might occur in some additionally complex, combined manner. Understanding that complex underlying associations may exist in your data is an important part of how you go about key elements of a biomedical data analysis pipeline, so that you don't miss these possible relationships. Now let's focus a bit more on the unique challenge presented by epistatic interactions. First off, note that there can be a difference between biological and statistical epistasis. Biological epistasis implies that there is a physical, pathway-based, or causal interaction with respect to outcome. Statistical epistasis is limited to an interaction between features that are predictive of outcome. This might happen independent of a biological interaction. However, we want to use statistical or machine learning methods that are capable of detecting statistical interactions that still may be essential to training an optimal model. Next, let's contrast multivariate effects in general with those that would be considered multivariate interactions. Let's start by looking at a simple multivariate additive effect, where each feature contributes its own univariate effect that adds up in how it impacts phenotype or outcome. In this illustration, we have two features, each with only two possible values, 0 or 1. Feature 1, in green, has its values represented in the plot by the colors red and black, and feature 2, in blue, has its values represented in the plot along the x-axis. The y-axis is the phenotype or outcome, which we're trying to predict. The 4 dots on the plot represent the phenotype value, so let's say something like patient height. So as a baseline, imagine that if neither feature had a relationship with phenotype, the phenotype would be the same for all of these points. First, we could imagine the univariate effect of feature 1 on the phenotype as being the difference of the average of the red dots versus the average of the black dots. Next, we could visualize the univariate effect of feature 2 on phenotype as the difference in the average of the left-sided dots from those on the right. What should be clear here is that both features have their own independent association with phenotype, and that these effects, adding up, are leading to a larger value of phenotype cumulatively. However, there's no interaction effect here. Now we'll look at a multivariate multiplicative effect. Again, we have the same setup where we can confirm that both feature 1 and feature 2 each have their own univariate association with outcome. However, overall, we can see that the combined effects of the 2 features on outcome leads to a greater change than the sum of the 2 individual effects. Therefore, this is considered to be an interaction effect. The impact of interactions on outcome can only be fully appreciated when the values of the features are considered together. Lastly, we look at a multivariate crossover effect. This is also referred to as a masking effect. Here, the effect of one feature on outcome can be partially or fully hidden by the other. Using our same example, we can see that for feature 1, the average phenotype of red versus black dots would be almost exactly the same. So if we had used a univariate test of association for feature 1, we might incorrectly think that feature 1 is irrelevant to predicting outcome. And we have a similar situation here for feature 2. However, what should be evident from this illustration is that despite outcome having no clear relationship with either individual feature, the specific combination of feature 1 and feature 2 values very much impacts whether the instance has a high or low phenotype value in the data. But we don't know ahead of time the true nature of complex multivariate associations going from one data set to the next. So it's important to consider these worst-case scenarios of data complexity when deciding what assumptions you're willing to make in setting up your own data analysis pipeline. Now let's dive a little deeper into how complex associations such as epistasis can make data analysis and machine learning more challenging. The curse of dimensionality refers to various phenomena that arise when organizing or analyzing data in high-dimensional spaces that don't occur in low-dimensional ones. Analyzing univariate effects of features in a dataset would be low-dimensional, but exploring all unique feature combinations would be a high-dimensional one. Consider an example such as conducting an exhaustive analysis of 500,000 genetic variants and their associations with outcome in a genome-wide association study, or GWAS. If we only looked at univariate associations, our search space would intuitively be 500,000 possibilities. However, what if we wanted to look at all univariate effects as well as all possible 2- and 3-way epistatic interactions among those 500,000 features? Now our search space has climbed to approximately 2.08 times 10 to the 16 possibilities, which happens to be 100 million times greater than the number of stars in the sky. Let's generously assume that your personal computer is able to complete 200 million search operations per second. That means that this search would take a minimum of 120 days to complete. Beyond computational burden, the curse of dimensionality also dictates that as we consider higher-order combinations of variables, we will observe exponentially fewer instances for each feature value combination. For example, here we see instances separated into quantitative regions, either as a single feature, a pair of features, or 3 features. Notice that as the dimensionality increases, the number of instances that end up in any partition of the overall problem space becomes more sparse. And this goes to the point where some or many partitions will have no instances at all. This phenomena directly impacts power, which is the probability of a hypothesis test finding an effect if there's one to be found. Essentially, searching in higher dimensions is the same as trying to find an effect with too small of a sample size. The point here is to make you aware that there are some limitations to applying traditional analytical approaches. And sometimes it's not practical to try and tackle an exhaustive search of the solution space, but instead to more cleverly and efficiently explore a subset of the search space. We'll end this discussion of epistasis by providing this example of a study that sought to look for epistatic interactions in a genome-wide association analysis of Alzheimer's disease. Feel free to check this out. Another set of complex associations we mentioned include heterogeneous associations, such as genetic heterogeneity and phenocopy. To understand this complicating phenomenon further, assume we had a group of patients that all had a disease outcome of interest, and that from a clinical perspective, the disease phenotype or phenotypes of these patients was either similar enough that we couldn't reliably distinguish them, or we observe a range of phenotypes, but stratifying patients by the severity of disease phenotype doesn't really help us to better model underlying associations. Either of these scenarios is possible when you're dealing with heterogeneous associations. Imagine then that under the surface, these patients actually belong to underlying subgroups, each with a unique set of features that predicted or caused their disease phenotype. Heterogeneous associations are also a likely contributor to what's known as missing heritability, or the unexplained genetic risk of a disease. And it's likely an important part of developing more effective personalized medicine. From a machine learning modeling perspective, genetic heterogeneity can be a difficult problem to overcome, since ideally, most algorithms seek to train a model that can be uniformly applied to predict outcome of all instances. Here, we look at a study considering heterogeneity and its relationship with improving personalized medicine. This study considers a group of patients with adenocarcinoma of the lung, otherwise known as lung cancer. The different color of each patient reflects heterogeneity according to the respective driver oncogene mutations they each possess. For example, EGFR is an oncogene that is known to be a predictor of response to a specific treatment called EGFR TKI, which is a tyrosine kinase inhibitor. The number of people in each heterogeneous group reflects the approximate incidence of each oncogene in the patient population. 90% of EGFR mutations, including L858R and exon 19 del, are known to be predictors of desirable treatment response. However, there is further heterogeneity in patients with specific EGFR mutations, and these patients had varied response to the EGFR TKI treatment. These other heterogeneous predictors of inherent resistance to treatment typically lead to shorter survival outcomes. And beyond the patient group as a whole, there's further heterogeneity in individual patients treated using targeted therapy that results from the selection pressure and microenvironment of tumors. So in this case, treatment could actually exacerbate the heterogeneity of tumors in a given patient. Stepping back, this whole example is further meant to emphasize the unique challenges of biomedical data problems. They can be noisy, large-scale, complex, and have high stakes when it comes to decision-making. If you want to read more about genetic heterogeneity, I welcome you to check out this review that I co-authored covering challenges, impacts, and methods for considering heterogeneous associations. And here, you can find an example research paper seeking to develop and apply a machine learning method that can directly capture heterogeneous as well as epistatic associations. So, to summarize this lecture, the modern era of biomedical data analytics now includes focus on large-scale or big and multimodal datasets. Also, there's value in considering and integrating both traditional hypothesis-driven experimentation as well as newer data-driven or hypothesis-generating experimentation approaches. There are also myriad sources, formats, types, and scales of biomedical or clinical data. And there are many challenges to overcome in preparing and analyzing these data types. Overall, there's a clear need to develop and apply more sophisticated, flexible, reliable, transparent, and efficient computational approaches to tackle and take advantage of increasingly big biomedical data in all of its forms. I'm Ryan Urbanowitz, and thanks for joining me for this lecture. Stay skeptical, curious, and creative.